Our next storyteller is Leonor Leandro. Science Takes a Village.
So it's midnight. I'm sitting in the cold basement of the plant pathology greenhouses here on campus. I am surrounded by growth chambers, dew chambers, incubators, and other machines that make this whirring sound that isolates me from the world around me. I carefully remove a strawberry plant from a dew chamber and put it on a wooden desk. I then punch two small discs out of the leaves, place them on a drop of fixative on a glass slide, put a drop of stain on top, and cover them with a cover slip. I will later be observing these specimens under the microscope. At the time, I was a second-year PhD student conducting research on strawberry anthracnose. It's a fungal disease that causes these brown sunken lesions on the fruits. In this experiment I'm conducting, I'm trying to understand if the pathogen called Colletotricum acutatum can survive and sporulate on the surface of green strawberry leaves, and how the periods of dew affect those processes. The experiment involves applying the fungal spores to the leaf surfaces, incubating the plants in dew chambers, and then taking leaf samples in a very intensive sampling schedule, every 3 hours for the first 24 hours, and then at 36 hours, 48, and then every 24 hours for another 5 days. 12:30 a.m. As I sample the last leaf discs, a friend sits in a chair, half asleep. I had begged him to come with me to the greenhouse. I didn't want to be down there alone at that time of night, and I needed his help for the sake of science. He had agreed after a promise of a free pizza dinner. I was so thankful for his help. I was able to do my work without being distracted by the creepiness of the place. 3 a.m. I lay awake in bed. My advisor is hard at work in that cold greenhouse basement. He offered to take the 3 a.m. sample for me, for my mental sanity, and also for scientific rigor. I was really lucky to have such a supportive advisor. I had heard of colleagues who had similar experiments that had slept in the lab or in their offices to do similar work. I had hesitated to take my advisor's help, but we both knew it was for the best. And there was no way I was sleeping in that greenhouse basement. But I couldn't help worry. What if he does not do it right? What if he mixes up the samples? What if he completely forgot to go there? Gulp. 5:30 a.m. I drag myself out of bed and to the greenhouse. It's still dark out, and I would rather have someone with me, but it wasn't quite as easy to convince a friend to come with me at that hour in the morning. I walk down the stairs to the basement, and as I turn on the light, a large, brown, buzzing insect flies right towards me, barely missing my head. Those pesky cockroaches. I get to work and collect my samples. 9 a.m., I take the last sample of that intensive 3-hour interval sampling. The worst is over, so I thought. The next day, I sit excited at the microscope. I am ready to observe my samples and count the number of spores that had germinated, produced aprosoria, those dark round structures that serve for attachment and survival, and the number of spores that had produced additional spores on the leaf surface. I peer through the microscope lens and focus on the spores. There they were, stained blue on this light green background of the leaves. But something was wrong. The spores were just sitting there, ungerminated, dormant, or dead. What could have happened? Before this day, I had been carefully planning my experiments, and I had made sure the fungal cultures were clean, the strawberries had green leaves, and I had also run a short preliminary experiment in that growth chamber to make sure the settings were right and the spores were germinating. Everything had worked perfectly. So I look at another sample and then another to check if it wasn't just a fluke. But no, none of the spores had germinated. My heart sank. In the days that followed, I spent hours brainstorming with my advisor, my co-advisor, my lab mates, even my mom, about what could have gone wrong. Was the fungal culture infected by a virus? Had the plants been sprayed with a fungicide in the greenhouse? Did the chamber overheat and kill my spores? I tested each of these hypotheses one by one, but couldn't find an answer. Colleagues would stop me in the corridor and pose more hypotheses. Were the lights on too long in the chamber? Could it have something to do with the phase of the moon? Weeks went by, and I started to panic. This was seriously delaying my research progress. These experiments were an essential part of my dissertation. If I couldn't get them working, I would never graduate. I couldn't get my PhD. Should I quit? I was clearly a failure. One day, my co-advisor asked me, "What water did you use to inoculate the leaves?" I said, "Well, sterile distilled water, of course. I know that golden rule." And then he asked, "But what water did you use to create dew in the chambers?" And a light bulb went on in my head. The chambers did use a different source of water. So this is how it works. Picture a green, lush strawberry field at dawn. The air feels cool, and the sun has a warm orange glow to it. As you walk in the field, your shoes get a little wet. You look down, and you notice all these little droplets of water on the plants. Morning dew. Dew chambers recreate morning dew, but in a clunky, mechanical kind of way. Imagine a white chamber that towers above me, metallic chamber. On the outside, there are buttons and knobs and little lights that turn red and green depending on the cooling or heating cycle. On the inside, there are cold metal walls, metal shelves, and a large metal tray on the bottom. That tray is filled with water, and it has a coil that heats that water. So as the water evaporates, it reaches the cold walls and the cold surfaces of the leaves, creating morning dew. That tray at the bottom of the chambers is connected to city water, Ames city water. So could that be the problem? If so, why did it work in my preliminary experiments? Then everything made sense. That first time when I ran the preliminary experiment, the water had been sitting at that tray for weeks, even months. And I knew that because the water was green from algal overgrowth, and who knows what else was lurking in there. So I had decided that I should clean out the tray, disinfect it, and fill it with fresh tap water. Do you know what's in fresh tap water that's not in water that has been sitting out for a while? Chlorine. And do you know what we use as a primary disinfectant against microbes? Chlorine. So it was hard to believe that water that we drink every day here in Ames would have enough chlorine to kill my fungal spores. But after conducting several germination tests, I showed that was the case. The minute amount of 2.7 parts per million of chlorine in Ames tap water was killing my Colletotricum spores. With this new information, I was able to get back on track on my PhD. I conducted several runs of my experiments and gained significant insights on germination and survival of Colletotricum acutatum on leaves and how the cycle of this disease works. I was able to graduate, publish my papers, and eventually get a faculty job. So although this story happened many years ago, I've never forgotten an important lesson. First, don't use tap water in your experiments. And second, it takes a village to do good science. I couldn't have made it through those obstacles in my PhD without the excellent guidance and support of my major professor, Dr. Mark Gleason, the insights from my co-advisor, Dr. Forrest Nutter, the kindness of the friends who stayed with me in the greenhouse basement, and the support of many other family and friends. I have now been a faculty member at ISU for 12 years, and I've continued to apply that lesson in the way I mentor my students, run my research program, and interact with colleagues and those around me. Thank you for listening.